Hello, everyone. I first would like to thank the organizing committee for the opportunity to participate in this very important Congress. We're going to discuss some aspects related to nutritional strategies and feeding strategies associated with helping cows maintain optimal health and production during the transition period. Now, the way in which I've structured this presentation, it's one in which I want to emphasize work that has been done in the last 10 years or so regarding specific aspects of feeding cows during the transition period, either with specific nutrients such as choline, methionine, trace minerals, but also emphasize some new approaches, if you will, that we're starting to see more publications about in the context of feeding cows around parturition. One clear example of that approach is the continued increase in the use of microbial fermentation products. We'll talk a little bit about some of the available data. And also I'm going to emphasize what I consider to be some up and coming novel approaches to feeding cows around parturition to help them maintain optimal health. And that has to do with the use of specific essential amino acids. So we're going to talk a little bit about the role of lysine and the nutrition of cows around parturition, and also emphasize how not only the metabolism of lysine, but potentially other nutrients that are associated with the so-called one carbon metabolism pathway can help generate other molecules such as carnitine and potentially help cows maintain optimal health. As you can see from this summary, over the last 10 years or so, there's been quite a bit of work associated with transition cow nutrition and feeding. And out of those papers, which are close to about 280, 290 papers published in PubMed, there's been quite a bit of work, for example, on choline and methionine. We've also seen quite a bit of work in regards to calcium metabolism and really maintaining cation-anion difference. So we're going to talk a little bit more about how those papers have generated new data, or how those papers have emphasized how the management of cows with some of these nutrients consistently improves health and performance. Now, but before we get into the details of specific details related to feeding cows around parturition, I want to spend a few minutes discussing the environment of the cow around parturition, meaning not only metabolism, but also talking a little bit about the endocrine changes that occur and the immune changes that occur around parturition that somewhat dictate the approaches that we need to take in the context of nutritional management. Now, around parturition, we're all familiar with the gradual decrease in feed intake that occurs. This is a normal process. Of course, that decrease in feed intake means that cows are going to enter a period of negative energy balance, which we see here decreasing from that value of 0, which would indicate a net balance in energy in the cow. So this is something that we've seen for many years, and of course this is one aspect that we've tried to manage, meaning we've tried to alleviate that very negative change in energy balance in the cow through nutrition. Now, something else I want to highlight here is the fact that if we were to achieve optimal performance or optimal health in cows around parturition, ideally we would, for example, keep calcemia at 100%, okay, and immune competence also at 100%, but that's really not what we see in vivo. Cows are going to, again, gradually experience a period of immune dysfunction around parturition, and this effect, it starts going back to normal, if you will, as a cow progresses into lactation. Now, immune competence obviously is very important, because around parturition is when cows are going to be most susceptible to experiencing some type of either metabolic or pathogenic disorder. Now, associated with immune competence is this transient period in which the animal actually experiences an inflammatory state and an inflammatory state that's actually related to also a period of oxidative stress. These two processes are linked. And again, ideally, we would like to keep those at a very low level because from a nutritional standpoint, they also plays a toll on nutrient utilization by different systems of the cow besides the mammary gland. So we're going to talk a little bit about how these physiological and endocrine changes around parturition affect the nutrition of the cow and what we can do to try to alleviate some of those issues. Now, I want to remind you that during pregnancy or towards late pregnancy, remember, we're still feeding the cow not only to ensure that she makes a good transition into lactation, but we're still feeding not only the fetus, which is entering those final stages of growth, but also the uterine tissue, which needs to help that fetus get to the point in which it's ready to hit the ground, if you will. So again, we're not going to talk about how nutrition affects the fetus itself, but this is also an important area of research that you're going to see more data being published in the next few years. How the nutrition of the cow impacts the growth and performance of that calf. Now, in terms of the adaptations of the cows, again, we looked at some of the major physiological and endocrine changes that occur around parturition, but I want to discuss a little bit in more detail what happens at the tissue level and at the cellular level. Now, again, feed intake or a decrease in feed intake around parturition is a normal response of the animal. A lot of that has to do with the endocrine changes that occur. For example, pro-inflammatory cytokine production is increased right prior to parturition. They contribute to signaling in the brain that tells the cow, you need to stop intake or consuming feed. Again, that's a normal type of response that occurs right before parturition. There are other things that can potentially affect the ability of that animal to eat optimally, and that has to do, for example, with concentrated forage level in the diet. Again, we're not going to discuss those aspects to any large extent, but they can definitely, for example, increase rumen field, and again, that in itself is going to prevent cows from reaching optimal intakes. Body condition, cow comfort are important aspects that need to be taken into account when it comes to optimal feed intake. But again, assuming that we can manage all of those aspects reasonably well with available information, let's just focus on we're going to focus only on how specific nutrients can actually help some of these changes that occur around parturition. Now, remember that even when the cow is reducing feed intake after parturition, there's this normal response of the animal to start eating more feed. Part of that has to do with helping the mammary gland meet its requirements for nutrients. Of course, you're going to see as the animal increases feed intake after parturition, you're going to increase in the size of the rumen. We're going to have a lot of adaptations at the level of microorganisms because we often go from different diets between late prepartum and the early postpartum. Of course, as the rumen tissue develops, the absorptive capacity of that organ is going to increase, and that's what's going to be important in the context of delivering volatile fatty acids to that animal. But remember that during the first week or two, or potentially even longer than that after parturition, there's going to be this gradual increase in feed intake. So potentially the supply of EFAs is going to be somewhat lower than optimal for those cows. So that in itself is going to trigger some other adaptations at the level of different tissues. Now, in addition to these changes that we see at the level of the tissue and in the context of the microorganisms, we have to keep in mind that there's often a transient period during which permeability of the rumen, it's also going to be increased around, right after parturition, meaning that potentially there's going to be leakage of ruminal contents. And by that, we mean potentially toxins such as lipopolysaccharides, maybe some other bioactive molecules that can actually enter the circulation in the animal. And those are potentially also effectors of changes at the level of other tissues. Now, those other tissues could potentially be the adipose and the mammary gland. Now, we've known for many years that one of the important adaptations that the cows undergoes right after parturition is this very large increase in lipolysis, meaning that those fat stores that were accumulated during the dry period are going to start going through a cascade in which fatty acids are hydrolyzed from the triacylglycerol stores, and they're put into the circulation and can travel to different organs. The liver is one that we've studied quite a bit over the years because of obvious reasons. It's the main organ that produces glucose from propionate, for example, and from other gluconeogenic compounds such as amino acids and glycerol. So again, the emphasis on the liver in the context of adaptations to parturition have been very important. But keep in mind that, again, with the knowledge that we've accumulated in the last few years, now we know that, for example, molecules that may make it across that rumen epithelium because of the increase in permeability can potentially trigger immune responses at the level of the liver. And those immune responses are going to encompass, for example, the acute phase response, which means that the liver is going to start utilizing amino acids to make acute phase proteins, which have some very important physiological roles at the systemic levels. We will talk a little bit more about that towards the end of the presentation. Some of those compounds coming from the rumen because of the increased permeability can potentially also increase reactive oxygen species production, and that in itself can trigger an inflammatory response. So again, the liver has to handle not only the metabolic aspect of having to produce glucose, but potentially some of these other immune roles, if you will, that are associated with what's happening in other organs of the animal. Of course, one of the important metabolic aspects of the liver around parturition, it's the increase in oxidation or the increase in capture of fatty acids coming from adipose tissue to generate ATP and to produce ketone bodies, which can be used by other tissues as sources of fuels. However, we've known for quite a long time that NIFAs and hydroxybutyrate, for example, above a certain level actually can have a detrimental effect at the level of the immune system. And again, there's been quite a bit of work indicating that those immune cells, for example, the neutrophils, which are important in the innate immune response, are going to be fairly susceptible to high concentrations of NIFA and BHBA, and potentially that can inhibit not only the ability of those neutrophils to phagocytose pathogens, but the ability of those immune cells to actually respond to some other challenges. Okay, and by that I mean potentially an increase in an inflammatory cascade at the level of the mammary gland because of some pathogen that's able to enter those mammary epithelial cells and create this mastitic response. So again, the immune system it's going to be fairly susceptible to all these metabolic adaptations that are taking place, particularly right after parturition. Now, let's talk a little bit about what happens at the level of the mammary gland, because that's going to be fairly important in the context of some other nutritional adaptations that take place in the cow. Of course, the mammary gland it's going to be the key organ at parturition because of its main function in the synthesis of milk. Of course, the number of secretory cells is going to increase fairly dramatically. Organ size is going to increase fairly dramatically. And nutrient needs of that organ are also going to increase quite dramatically. So the mammary gland is clearly one of the most important organs after parturition in terms of driving a lot of these adaptations. Now, it is not surprising then that a lot of the emphasis that we've seen, not only in the last 10 years, but before for many decades in the context of parturition and mammary function, it's the huge drain that the mammary gland places on calcium before parturition, because it needs to synthesize colostrum, and colostrum takes up quite a bit of calcium in the context of what's available in the systemic circulation, but also other nutrients. And these other nutrients could be amino acids, phosphorus, for example. They could be antioxidants. And we're going to talk a little bit about some of those as we move through this presentation. So again, in the context of calcium, these changes that occur right around parturition in the context of making colostrum and synthesizing milk are fairly important in the context of calcium metabolism. Again, this has been a topic of study for many years. It is not surprising that we're still trying to study ways in which we can optimize calcium homeostasis in transition cows, especially after parturition. So we'll talk a little bit about what's been done in the last few years. At the context of the gut, obviously calcium absorption it's important. The mechanisms of calcium absorption from the lumen of the intestine into the circulation are somewhat complex. We're not going to discuss them to any large extent, but I do want to emphasize that the process of calcium absorption is one that obviously not only relies on availability of calcium in the lumen of the intestine, but also in the ability of vitamin D and other potential molecules that are important in the transport of calcium across those intestinal cells. So clearly, cows often are going to enter this transient period in which plasma calcium is going to decrease. Potentially, they can enter a subclinical hypocalcemic period, and you can see here some old data kind of showing how the patterns of calcium will gradually decrease around parturition, whether you have a cow that experiences an issue with milk fever or one that remains healthy. You can see the difference here. So typically, we believe that about 8 milligrams per deciliter of calcium is what cows need to maintain in the circulation to avoid a risk of developing milk fever after parturition. Now, there's been quite a bit of work done in this area over the years, not necessarily about feeding calcium, but about ways, for example, to create these mild metabolic acidosis prior to parturition that could be helpful in the context not only of enhancing production of vitamin D3 at the level of the kidney, but the responsiveness of that vitamin D3 at the level of the intestine to help those animals optimize calcium absorption. Also at the level of the bone to enhance resources of calcium, such that availability of calcium not only to the mammary gland but also the systemic level is maintained in a way that does not have a detrimental effect at the level of the animal. So acidifying feeds using commercial products, typically based on protein meals, is something that has become fairly common. And in fact, there are a couple of recent meta-analyses of the literature that really highlight the benefit of using these acidifying products before parturition. There's some interest also in trying to use them after calving, but again, most of the emphasis has been on again creating these mild metabolic acidosis prior to parturition to get those cows ready to respond to the changes in calcium metabolism after parturition. And again, this experiment, this meta-analysis that was done in 2009, published in 2019, clearly emphasizes how monitoring DCAT balance prior to parturition. You can do that, for example, by monitoring urine pH, can help those cows achieve optimal performance after parturition. And again, some of this data, I believe, is fairly well established, and I'm not going to discuss it too much more in the context of calcium metabolism. Now let's move on and start discussing other aspects related to the immune response. And again, I already mentioned the inflammatory status and the reactive oxygen, or the state of oxidative stress that cows experience right after parturition. These are processes that are normal, in the sense that they occur normally in cows after parturition. There are biologically important reasons why these are important. For example, reactive oxygen species production is important in the context of immune cells, and their ability to kill certain pathogens that may be detrimental to the animals. So again, we do want to have some of these inflammatory and oxidative stress responses take place after parturition, as a normal response in the animal to try to fight some challenges. And again, this could come from infectious agents. They can come from, for example, the fact that maybe there's some antioxidant deficiencies occurring at the level of the animal. Remember that in the process of colostrum synthesis, there's a lot of nutrients that go into that process, and potentially antioxidants, and we're going to talk about some of those, could also be depleted during that process. So again, this from a nutritional standpoint, this could be very important. Now, the process of parturition itself, and as I indicated before, the increase in metabolic demands and the production or the increase in concentrations of NEF and BHV are important factors contributing to the inflammatory and oxidative stress response. And even the length during which these processes remain elevated in the cow, which may not be a good thing. Of course, in the context of those processes, we need to think about antioxidants. And as I indicated before, the mammary gland is an important player in that, in the maintenance of proper antioxidant balance in the animal. We're going to talk about some other processes that the animal can use in certain tissues to generate some of these antioxidants endogenously. One other important aspect of parturition in the context of generating pro-oxidant molecules is the metabolism of polyunsaturated fatty acids, primarily in the adipose tissue compartment. Again, this is a process that right after parturition, when you have these catabolic states, potentially could lead to the production of molecules referred to as oxylipids. These oxylipids, again, work in fairly complex signaling pathways. We're not going to discuss them very much, but there's some evidence that has been published and emphasizing the potential for this metabolism of polyunsaturated fatty acids in tissues like adipose to generate these compounds that can contribute to the inflammatory and oxidative stress cascade. So not only diet or nutrition can potentially impact these immune type responses, but also the normal catabolism of the animal, specifically in the adipose, can contribute to that. So that's important to keep in mind. Now, in the context of metabolism and production of reactive oxygen species, which again become very important players in the context of this oxidant and antioxidant cascade, the liver, it's probably the most quantitatively important organ in which this process becomes fairly important. Now, a classical study from Chris Reynolds back in 2003 demonstrated that between late prepartum and early postpartum, the metabolic rate of the liver more than doubles. So there's a large increase in oxygen consumption by the organ. And of course, oxidative metabolism is the key contributor of reactive oxygen species at the level of the mitochondria. So again, the production of reactive oxygen species is a normal process that occurs as cows are going from late prepartum to early lactation. This is a process that is going to occur in all cows. The issue here is ensuring that we're making those cows capable of fighting the potential increase in reactive oxygen species that will occur at the level of the liver, for example, but cows can also face this in other metabolic important organs like the mammary gland. So the key is to help those cows be ready to potentially fight those very large increases in reactive oxygen species. Now, on the other side of that equation is the fact that leukocytes or phagocytic leukocytes, particularly those in the innate immune system, also rely in production of reactive oxygen species to effectively kill pathogens and potentially help the animals. So reactive oxygen species production is not only something that occurs at the tissue level, but again, it's a normal response of some of the immune cells specifically during this period right after parturition. So what do these reactive oxygen species do from the biological standpoint? These molecules, which again can be classified into different types, which we will not discuss in this presentation, can actually trigger an immune response, but also can trigger mechanisms that can help tissues and cells fight that inflammatory oxidant response. And I'm showing some of the key mediators of the pro-inflammatory response, such as NF-kappa beta, and a key antioxidant transcription regulator referred to as NRF2. These linkages between reactive oxygen species and these pro-inflammatory and antioxidant mediators have been very well studied in monogastric animals, not so much in ruminants. There's a growing body of data that's indicating that the bovine cells can also have functional signaling pathways associated with these responses. So one of the key questions that we need to answer from a nutritional standpoint is whether certain nutrients can actually trigger our response at the level of these mediators, and particularly the antioxidant response. Now, in that context, it's also important to keep in mind that cells and tissues have access to a number of antioxidant compounds that can be used when necessary. Of course, we can supply these antioxidant nutrients or compounds through the diet. One of the big issues with ruminants and particularly transition cows is understanding what the requirements for the various potential antioxidants are. And we simply do not have a very good handle on requirements. Again, that's a limitation. That's something that we need to, we're going to have to deal with in the future. We also need to understand that the requirements, especially around parturition, are going to change because of these changes that the animal goes through from late pregnancy, parturition, and early lactation. And on top of that, we're going to have requirements that are going to change at the level of specific organs and cells. So again, it's an important, it's a difficult question for us to try to address with a single experiment. That's why you see a lot of emphasis in trying to understand not only how certain amino acids can potentially aid in terms of providing endogenous antioxidants. We're going to talk a little bit more about that. But you see a lot of research trying to understand better how the supply of the classical antioxidants like certain vitamins, certain trace minerals helps cows around parturition. And in fact, we've known primarily from monogastric experiments, we've known for a number of years that nutrients like water-soluble vitamins, like vitamin A, vitamin C, are very important in the context of the antioxidant response. There are some trace minerals like selenium, copper, zinc that may not be directly considered radical scavengers, meaning that they can actually trap some of those reactive oxygen species and make them less deleterious to the system. But some of these trace minerals actually are cofactors for very important enzymes in the antioxidant cascades. Perhaps the most important or most studied, if you will, in that context is the activity of glutathione peroxidase, which requires selenium as a cofactor. In fact, glutathione peroxide along with glutathione reductase are very important in the context of the antioxidant capacity of many organs in the cow and in monogastric animals as well. The other important aspect of this particular enzymatic reaction is the need for glutathione. And we're going to talk a little bit more about glutathione and how we can potentially help those cows synthesize glutathione through other nutrients provided in the diet. But before we move into more discussion of that, let's talk a little bit about some of the available data linking trace minerals with health and performance in transition cows. There's not a lot of work specifically dealing with the supply of trace minerals or the type of trace minerals around parturition. Most of the studies that have been published deal with supplementation right after parturition and during early lactation, and that's where the data in this particular meta-analysis came from. Fairly large number of studies, and you can see here the type of amino acid complex trace mineral products that were fed. But overall, based on these meta-analyses, even though there's quite a bit of differences in terms of the response across studies, in general, supplementing these amino acid complex trace minerals had a beneficial effect in terms of milk production. So again, clearly trace minerals could have an important role in the context of the nutrition of cows around parturition. Some of these trace minerals, as I indicated in the previous slide, not only are important cofactors of enzymes, but some of these have effects at even more fundamental levels in terms of coordinating cellular apoptosis and cell differentiation. Again, it's the mechanisms whereby these trace minerals have an effect at the cell and tissue level could be quite complex. Now, in our group, we have done some work looking at replacing a portion of the sulfate or inorganic form of trace minerals with amino acid complex trace minerals in terms of performance and inflammatory state of cows around parturition. We published a few studies in that regard. And again, I'm showing here some of the performance data and biomarker data we were able to generate. Clearly, again, in this particular experiment, cows that were supplemented with the amino acid complex trace minerals had better rates of energy-corrected milk production, and you can see it here. Dry matter intake tended also to be greater in those cows. So again, it benefited both aspects both of the major aspects of production. And in the context of liver metabolism, for example, with incidence in the context of the acute phase response that I mentioned before, cows that were fed these amino acid complex trace minerals actually had a better response in terms of the synthesis of albumin. Albumin is an important acute phase protein that is produced around parturition. Typically, when you see a decrease in albumin in the circulation, that is an indicator that the animal is experiencing potentially an acute or chronic state of inflammation at the level of the liver. And what we were able to detect in this experiment is that, again, those cows that were fed with the amino acid complex trace minerals actually did better. They seem to be in a better immune and antioxidant state. And again, there were other markers like a lower concentration of IL-6, haptoglobin, and ceruloplasmin that, in addition to albumin, told us that in fact this amino acid complex supplementation had a beneficial effect in terms of the inflammatory and oxidative stress state of the animal. Again, working with trace minerals and trying to understand requirements and benefits, especially of products that can deliver more bioavailable sources, it's an area of active research. Okay, and again, it's difficult to try to understand actual requirements or needs of cows around parturition in the context of amino acid of trace minerals. And we happen to agree with the statement made by Overton and Jesui in the sense that what we're going to have to rely more often than not is on phenotypic changes or responses, not only at the level of production, but at the level of physiology, in the context of trying to understand whether certain trace mineral and or forms of trace minerals actually having a positive effect in the animal. And there's been quite a bit of work trying to understand, for example, or methods that could potentially enhance the bioavailability of these trace minerals. One example that I want to share with you that does not necessarily deal with transition cows, but it deals with a very important type of stress, which is thermal stress, is this experiment that was published in which cows were actually challenged through a heat chamber to a fairly high THI of 83. So what they did in this particular experiment was to try to understand whether replacing selenium selenide, which is a typical product that delivers selenium to animals, by selenium methionine would actually have a positive impact in the context of the antioxidant response and production. And clearly, what you can see here is that even though there was no difference in the context of production, when we look at the total amount of selenium that was detected in milk, clearly, the fact that there was a greater amount of total selenium in those cows fed selenium methionine during the heat stress challenge told us that in fact bioavailability of that selenium was greater. Now, what does that mean, or what did that mean in the context of the physiological response in the animal? We can see here a couple of pieces of data. One, the glutathione peroxidase activity on the top panel, and then the concentration of lipid peroxidation products on the lower panel. And you can clearly see that those animals fed the selenium methionine product were actually able to maintain a more consistent glutathione peroxidase activity during that heat stress challenge, and in fact had lower concentrations of lipid peroxidation products and even lower concentrations of hydrogen peroxide, while maintaining higher levels of a liver function enzyme, adenine aminotransferase, in the circulation. So again, this is an example in which the bioavailability of the trace mineral, it's obviously an important aspect when we're trying to decide whether we feed inorganic sources or potentially some other more bioavailable sources of trace minerals to cows. Now, let's move on to another important topic in the context of feeding transition cows, and it has to do with the use of methyl donors, primarily choline and methionine, in the nutritional management of cows. In the monogastric area, choline and methionine I consider not only essential nutrients, but also functional nutrients, meaning that they're able to elicit effects on functional aspects that go beyond, for example, the synthesis of proteins, as in the case of methionine. To understand how these two methyl donors are important from the functional aspect, we need to understand a little bit better the so-called one-carbon metabolism pathways. Now, there are three main pathways that compose one-carbon metabolism. So what we have here is the methionine cycle, an abbreviated form of the methionine cycle. Clearly, one of the main aspects of the methionine cycle is to remethylate homocysteine back to methionine, a way in which methionine can then contribute S-adenosylmethionine, which is an important methyl donor. For our purposes, remember that the production of phosphatidylcholine, which is important in the synthesis of VLDL, is one that requires S-adenosylmethionine. Okay, so this methionine cycle is important in the context of, again, very important metabolic aspect of the liver, for example, which is the export of lipoproteins through VLDL. The other important thing that needs to be kept in mind when we look at the methionine cycle is the fact that at least two of the key enzymes, BHMT and SAHH, and we're going to talk a little bit more about BHMT towards the end of the presentation, require trace minerals as cofactors. So methionine cycle, it's obviously important in the context of the need for methionine to drive the cycle. But also, we need to keep in mind that regulation of this pathway it's more complex because of the need for certain trace minerals for optimal activity of these enzymes. And I emphasize this because towards the end of the presentation, we're going to discuss a little bit more in more detail metabolism of methionine and choline. The other important pathway in this one-carbon metabolism cycle is the folate cycle. And as the name implies, this has to do with availability of folic acid and a number of metabolic reactions that need to occur. I want to highlight that several enzymes in this pathway also require cofactors, and in this case, water-soluble vitamins, B12 and B6, and you can see them here. The other important pathway in this one-carbon metabolism pathway is the transsulfuration pathway. This is one which is very important in the context of the endogenous synthesis of antioxidants, specifically glutathione and taurine. The key enzyme here is cystathionine beta synthase, abbreviated as CBS, which again has as a requirement vitamin B6. So as you can see, the metabolism or the functioning of this one-carbon metabolism pathway requires a number of substrates and a number of cofactors for optimal activity. So even though we talk about methyl donors in the context of methionine supply, choline supply, betaine supply, or even folic acid supply, we need to keep in mind that there are a lot more complex requirements for trace nutrients that will have to be taken into account for an optimal functioning of this cycle. The key I do want you to keep in mind as we move forward in the presentation is the role of glutathione in a number of cellular processes across different tissues. In fact, glutathione is very well studied in non-ruminants as one very important player in the context of the antioxidant cascade. And I made the point earlier when we were talking about selenium and glutathione peroxidase that this reaction requires glutathione. So production of glutathione, it's an important aspect of the antioxidant response of cells and tissues. In monogastrics, there are a number of things that can affect production of glutathione. Not surprisingly, negative nitrogen balance is one important aspect that will decrease uh, glutathione synthesis. Starvation is another one. Inflammation can obviously also overcome that functioning of the transsulfuration pathway, and again, can potentially have a negative impact on the synthesis of glutathione. So this is important for you to keep in mind as we move forward and discuss a little bit more the emphasis or the interest that we've had in dairy nutrition on methyl donors, specifically methionine and choline. And in fact, the interest in these two nutrients in the context of dairy cow health it has been around for a very long time, many decades. I show here, for example, on the left, the first paper in which methionine was actually discussed as one potential nutrient that is deficient or that can lead to cows developing ketosis. Okay, on the right of this slide, I have some data that was again generated many years ago, in which researchers try to understand how choline or the metabolism of choline, for example, by comparing monogastrics versus ruminants, could actually have an impact on the production of very low density lipoproteins, which at the time was recognized as one potential pathway that could be affected negatively in cows right after parturition and lead them not only into developing of ketosis but also fatty liver. So again, the concept that methionine and choline can have an important metabolic function in specifically in the liver of ruminants is one that's been around for a long time. Now let's talk a little bit about the role of methionine supply during the transition period. And then we'll talk a little bit about data that's available for the role of choline around parturition. Again, these nutrients are not only important in the context of liver health, but we're going to talk about how those nutrients also benefit production. Now, in the context of methionine, again, you saw some very old data in which there was an association between the methionine deficiency and potentially ketosis or developing of ketosis. Over the years, there's been other experiments that have generated data indicating that, for example, there are negative associations between circulating concentrations of methionine and the incidence of lipidosis, for example. And again, that it's important to keep in mind that in the context of liver-related health problems, remember what occurs between late pregnancy and early lactation in the context of metabolism. Oxidative metabolism increases quite dramatically, and that in itself is going to increase the amount of reactive oxygen species that are produced. So potentially methionine availability has an impact in the context of the antioxidant response in the liver after parturition. One other interesting thing about methionine is some data that indicated that out of all the essential amino acids, the liver actually had a net extraction of methionine and histidine, in fact, between late prepartum and early lactation. So again, these were fairly novel data generated using multi-catheterized cows to try to understand how, for example, the liver metabolizes essential amino acids around parturition. To us, the fact that the liver had a net uptake of methionine after parturition meant that potentially methionine is not only needed for synthesis of protein in the liver after parturition, but potentially for the synthesis of antioxidants. And that's one of the pieces of data that we use to develop the hypothesis that in fact glutathione synthesis right after parturition is an important fate of methionine metabolism in the liver. So we designed a series of experiments to try to understand if in fact the methionine supply through either rumen protected forms or hydroxy analogs could affect antioxidant production in the liver. In the process of studying those aspects of one carbon metabolism, we also measured or generated the first measurements of the activity of three very important enzymes that I indicated or highlighted in one of the previous slides. BHMT, methionine synthase, MTR, and cystathionine beta synthase, CBS. So those are obviously key enzymes in each of these pathways that I mentioned belong to one carbon metabolism. And the data that we have generated across studies that we have published clearly indicates that first, there is an increase in the activity of all these enzymes between late prepartum and early lactation. So again, this activity of these enzymes or flux, potentially the flux through this one carbon metabolism increases after parturition. So the key is to try to understand why that occurs and what that means to the cow. We happen to believe, based on data that we have generated, that, for example, the marked increase in cystathionine beta synthase after a parturition is one of the homeoretic mechanisms whereby the liver generates additional concentrations of antioxidants to try to prevent issues with oxidative stress at the level of the liver. Based on that hypothesis, then we try to understand how the supply of not only methionine but also choline potentially can change the activity of these enzymes and what that means, not only in terms of antioxidant synthesis, but in terms of the synthesis of other intermediary compounds in those pathways that could potentially have a beneficial effect at the level of the liver. And I'm talking specifically, for example, about carnitine synthesis. We will talk a little bit more about that towards the end of the presentation. Just to show you a little bit, a summary of the experiments that we conducted in our group in the context of either feeding rumen protected methionine or hydroxy analog. Again, we've had fairly consistent responses in the context of dry matter intake, not only prepartum, but also postpartum. When we first started working in this area, we did not necessarily expect to see these increases in dry matter intake. So these were unexpected type results. But as I'm going to explain in the next few slides, we think we know why the animal is responding in that fashion when we increase the supply or the post-ruminal supply of methionine. And when we're talking about the post-ruminal supply of methionine, we're talking about levels in the metabolizable protein anywhere from 29 to about 33 in the prepartum diet to about to between 40 and about 56 grams in the postpartum diet. So roughly 7 to 8 grams of actually digestible methionine in the prepartum to about 15 to 16 grams of digestible methionine in the postpartum diet. And again, depending on the type of protected methionine or hydroxy analog that you're choosing to feed, you're going to have to figure out or determine how to get to these dosages that we have shown have beneficial effects. So again, not only in the context of dry matter intake, but also in the context of performance at the level of milk yield and also energy corrected milk. Again, these are consistent responses that we have been able to achieve. Now, there are other studies in which this increase in the supply of methionine has not necessarily impacted production. For example, the study of TOTS in last year, not only it actually verified that, again, multiparous cows, protected methionine actually has a positive effect in terms of energy fat-corrected milk. So again, that kind of supported the data that we had generated before, but in fact wasn't able to detect benefits at the level of production in primiparous cows. So again, probably the first experiment in which parity was one factor that they were trying to study in the context of the response to protected methionine. So again, depending on parity, when you're either managing your herd, or if you're designing an experiment, depending on parity, you may or may not see a response. But it's really a little bit more complicated than that, because again, it could the response to protected methionine could be related to the amount of other essential amino acids that you're supplying by the bioavailability at the level of the intestine, uh, and of course the source of protected methionine or hydroxy analog that you're feeding. Again, these are things that we have not necessarily tested, but you need to keep in mind when you're trying to either design experiments or design diets that you're going to feed out in the field. You need to make sure that you have sources that will allow you to deliver the post-ruminal level of methionine that can have a positive impact. Now, there are other environmental factors that could potentially interact with all these things that I'm mentioning in this particular slide, thermal environment being one of them. So again, there are many reasons, potential reasons why we may not see responses or consistent responses when we're trying to increase the post-ruminal supply of methionine. Again, this it's not an easy question to try to address, but it's important that we're aware of some of the data that have been generated out in the field. Now, from the context or going beyond production, strictly production aspects of feeding methyl donors, we need to highlight the fact that there's been a consistent response in the context of the role of methionine, or the beneficial effect of methionine on the immune system. And again, not only in our research group, we've been able to consistently detect decreases in inflammatory cytokines, for example, decreases in positive acute phase proteins like haptoglobin, which tell us that methionine actually is having a positive impact on the immune system, but also somewhat consistent responses at the level of methionine, also reducing the amount of reactive oxygen species in the circulation. Again, not only us, but other groups working in this area of protected amino acid supply with transition cows have been able to report some of the same findings. So again, the supply of methionine seems to have a positive effect beyond just production. And this is an important aspect of when it comes to trying to understand how to incorporate methionine into this overall picture of managing the cow for health and also production. One of the questions that we've tried to understand is what exactly is controlling one carbon metabolism, specifically these enzymes that I indicated previously around parturition. Is it the supply of methionine, the supply of choline? Is it potential? We haven't tried to address the question of trace minerals, but potentially those can also be part of these regulatory mechanisms. But what we've detected consistently is that, for example, in the case of CBAs, which is important in the context of synthesis of antioxidants, we've detected this very large increase after parturition. Now, we took all the data that we had available across 3 different studies in the context of enzyme activity and dry matter intake, plus other performance outputs, and try to run correlation analysis to try to see what kind of associations we could detect that may help us explain what's the regulatory mechanisms whereby activity of these enzymes it's controlled. What I'm showing here is the correlation between dry matter intake and CBS activity, and you can see that even though the correlation is somewhat low, it was fairly highly significant, meaning that dry matter intake actually had a beneficial effect or a positive effect in terms of pushing the activity of that enzyme, which, again, if we remember, CBS is important in the context of the use of methionine, potentially choline, all the way through different steps to synthesize glutathione and taurine. If we remember that, we can see how maintenance of dry matter intake or proper dry matter intake prior to parturition and after parturition, it's also going to potentially feed into this antioxidant capacity associated with the transsulfuration pathway. The interesting thing about this correlation analysis that we did is that when we try to look at the activity of something like BHMT, which is important in choline metabolism and methionine synthase, we couldn't find correlations. And in fact, we found a negative correlation between dry matter intake and methionine synthase that could help us understand a little bit better how these enzymes or these pathways are regulated. The regulation is probably a lot more complex. It could be at the level of gene expression. It could be at the level of the post-translational level. It could be associated with changes in the velocity of the reactions. Again, it's a complex type of regulation, and it's one in which I think we need to devote a little bit more effort to try to understand better, because at the end we might be able to link it with again a practical way of managing cows, and an example here is obviously methionine. Now, we've been able to detect increases in CBS activity, in particular in the context of supply of methionine. Our last experiment looking at this, the methionine supplied actually allowed us to detect a 20% difference in terms of CBS activity, which also translated into greater concentrations of glutathione in the liver. So again, this is an example linking an increase in the supply of methionine feeding into the methionine cycle and potentially through homocysteine into the transsulfuration pathway, and having a positive effect in terms of the antioxidant capacity of the tissue. Now, talking a little bit about work that has been done in the context of choline supply during the transition period. Again, there's been a lot of interest in trying to feed choline in the context of VLDL metabolism. Again, historically, this has been a very important area of study. Again, we know that choline can fit into the one carbon metabolism and potentially can lead to the generation of antioxidants such as taurine and glutathione through all those relationships that we have seen before. A recent meta-analysis actually of the available literature actually concluded that in the order of 12.9 to 13 grams per day of choline ion would benefit or would be optimal to benefit production. But an important question that we've had is, okay, well, what is really the optimal dose of choline ion that we should be feeding? And again, before we get into that, into an experiment that we have conducted, I wanted to show some results of this meta-analysis. And in fact, there's been two of those. The first one published in 2019, the second one in 2020. Both meta-analyses really concluded that there's a beneficial effect in terms of dry matter intake and milk yield when the supply of choline is increased around parturition. And you can clearly see here that the changes in the increases in dry matter intake, for example, tend to be the greatest between 10 to 20 grams of supplemental rumen-protected choline. That's what we have here in the top panel. In the lower panel, we can also see that most of the positive responses are really observed in between 10 to 20 grams of supplemental rumen-protected choline. So again, that's where this conclusion from the meta-analysis about 12.9 grams per day came from. So one of the other important aspects of this meta-analysis is that there was no consistent effect when all these data was put together in the context of rumen-protected choline alleviating liver triglyceride content, or on alleviating some of the inflammatory and antioxidant effects that occur during the transition period. So again, there's seems to be a clear effect at the level of production, but less consistent effects at the level of the immune and the antioxidant response. Now, we've tried to answer the question as to how much choline cows need. And again, we've used a feed restriction approach to try to create a severe state of negative energy balance. Remember that right after parturition, cows enter a fairly severe state of negative energy balance. We try to do that in mid-lactation cows. So we feed restricted those cows. And as you can see, we reach levels of negative energy balance fairly similar to what happens in cows right after parturition. So we tested 3 different levels of choline ion at the abomasum: 6.25, 12.5, and 25. And what we can gather from this information is that in terms of fat-corrected milk, again, there was a quadratic effect, and clearly indicating that potentially the 12.5 grams of post-ruminal supply of choline probably it's the optimal in terms of enhancing performance. And again, we have to keep in mind that the responses that we observed in this experiment, not only at the level of milk yield, but fat-corrected milk, were fairly similar that what has been reported in the in transition cow studies. The more physiological level, we can also see that, for example, choline supply had a linear effect in the context of reducing triglyceride concentrations. Again, this was expected. And in the context of phosphatidylcholine, we also saw a quadratic response, again, indicating that choline supply potentially can have an important effect, not only at the level of production, but at the level of metabolism. And again, the optimal dose based on our data seems to be somewhere between 6.25 and 12.5. Again, that's important to keep in mind. At the more mechanistic level, in this particular experiment, we also try to understand how the supply of choline could potentially alter 1-carbon metabolism. And we looked at, for example, at activity of VHMT. And what I'm emphasizing here in that orange box is the fact that there was a linear increase in the activity of this enzyme. And remember, 12.5 is sort of the optimal dose that has come out as the one that will create the most benefit in terms of feeding rumen-protected choline. But in fact, our data seems to support that conclusion. Now, remember, the activity of VHMT is linked to the metabolism or oxidation of choline to betaine, and then betaine to dimethylglycine. And in fact, when we looked at metabolic profiles in liver tissue, not only was activity of VHMT affected by the supply of choline, but there was a linear relationship between choline in the liver and betaine, meaning that the extra choline that was supplied during these abomasal infusions was actually being metabolized to betaine, and potentially that increased remethylation of homocysteine to methionine. When that happens, we can also expect that there will be more methionine being metabolized to the methionine cycle to S-adenosylmethionine, which in turn would probably help synthesis of phosphatidylcholine and VLDL, and explain why we found lower concentrations of triglyceride in the liver. Now, when we looked at the antioxidant response, if you will, through the activity of CBS, we actually saw a very interesting effect. And in fact, we saw a negative response in the activity as the amount of post-ruminal choline supply increase. That told us that potentially homocysteine was not being utilized, or metabolism of choline was not being used for the synthesis, or the metabolism of choline towards synthesis of antioxidant was somewhat reduced. And in fact, when we looked at carnitine levels in those cows, we also detected an increase in the concentration of carnitine, which, when we put everything together, led us to the suggestion that, again, homocysteine was probably not being used for the synthesis of antioxidants, but rather for remethylation of methionine. So these changes that we see here sort of agree with the previous slide, in which it would seem that as the concentration of intestinal choline increased, there was more of that choline being used to remethylate homocysteine, cysteine, again, to generate S-adenosylmethionine. And again, S-adenosylmethionine not only is important for phosphatidylcholine and VLDL synthesis, but can also provide methyl donors for synthesis of carnitine using lysine. So again, a number of important mechanistic aspects that we were able to uncover doing this particular experiment. And that moves us into another very important topic, and one that is I think it's a hot topic in the context of nutritional management of transition cows, and that's the supply of essential amino acids around parturition. Clearly, when you have cows that are not eating optimally, their essential amino acid supply is also going to be a limiting factor in terms of optimal milk and milk protein synthesis. Now, there's data out there that using abomasal infusions clearly showing that if the essential amino acid supply of cows is increased right after parturition, we're gonna see increases in the context of milk production, and this is what they were able to see in this particular experiment. Now, keep in mind that essential amino acids, or amino acids, are not only important for the synthesis of milk proteins and other blood proteins, but can also provide the backbone for other compounds. And again, I already made the point about methionine being used for the synthesis of homocysteine, and then that compound going through the transsulfuration pathway. Well, in the case of lysine is actually an important. It's the backbone for the synthesis of trimethyl lysine, which again will enter the endogenous carnitine synthesis pathway, which is actually very active in the liver. So theoretically, S-adenosylmethionine that is produced during the metabolism of choline, for example, or the metabolism of methionine, can furnish methyl groups for the synthesis of trimethyl lysine and then carnitine. And obviously, this would be important because historically, we've put some emphasis on the availability of carnitine in the context of enhancing fatty acid oxidation in the liver to reduce triglyceride synthesis. And in fact, there's one experiment that actually showed that that carnitine supply in the diet was effective in reducing liver triglyceride and increasing fatty acid oxidation without really affecting production. So again, carnitine supply would likely help cows maintain better liver health and overall health, but whether that's going to translate into better production, obviously it's difficult to tell from a single experiment. Now, there's more recent research done looking at lysine supplied around parturition. This is from an experiment that was published in 2019. In this particular experiment, one of the take-home messages was that, again, the supply of lysine prior to parturition had a beneficial effect on the level of feed intake, and also help those cows maintain lower concentrations of free fatty acids in the circulation, which again it's a reflection of potentially better handling of fatty acids at the level of the liver in the context of oxidation. If in fact that extra lysine or a portion of the extra lysine was metabolized to carnitine, or potential aspects at the level of reducing lipolysis because of the better intake. Now there weren't too many other measurements done at the level of the plasma or at the level of the liver to try to understand if in fact the extra supply of lysine increased carnitine. But again, potentially there might have been some of those effects in this particular experiment. Another recent experiment, in fact, published last year, also try to understand whether lysine supply or an increase in lysine supply pre and postpartum would have a beneficial effect. I'm only summarizing here some of the significant data that were generated, essentially establishing that greater post-ruminal supply of lysine prepartum really had most of the beneficial effects at the level of performance and in terms of some of the indicators of metabolism. Again, even though there was a tendency for better intakes, clearly there was a beneficial effect of lysine in the prepartum diet, or rumen-protected lysine in the prepartum diet in the context of enhancing energy-corrected milk. And in the context of plasma hydroxybutyrate, again, a tendency for lower concentrations, which essentially means that if we take into account the previous experiment, that again, lysine supply around parturition and especially prepartum could be something that we can fine tune in the diets of cows to try to help them remain in better health and obviously remain more productive. Now, moving towards the end of the presentation, I want to just touch upon some of the work that's increasingly being done in trying to understand how microbial fermentation products can actually have an important impact in the context of managing the nutrition of dairy cows around parturition. As you can see, what I have here is most of the papers that have been published, at least in General Dairy Science, in using different microbial fermentation products to try to understand potential benefits. One thing that that's kind of obvious when you analyze all these work is that there's really no consistent responses at the level of production. There's one experiment that seems to indicate that potentially there might be a dose response associated with production. Again, we can't really say much more out of a single experiment. What seems to be consistent, however, is the fact that there's some biomarkers of rumen function and health that actually have been improved, if you will, across some of these experiments. So again, these microbial fermentation products potentially, because of the fact that they contain antioxidants, vitamins, compounds like polyphenols, which are known to prevent inflammatory responses and help relieve oxidative stress. Potentially, those compounds are having this effect. Again, not only at the level of the rumen, but potentially at the systemic level. And again, there's one fairly new experiment in which it was demonstrated that these microbial fermentation products potentially can change the profiles of certain microbes in the rumen, which would have a potential beneficial effect in terms of rumen function. So again, there's a lot more work that I think needs to be done in this area of microbial fermentation products and their benefits. We still don't know the mode of action. We still don't know if we should be focusing on a certain molecule that's produced as part of these fermentation processes. So I think there's a lot of work that needs to be done in this area. Now, to conclude, I want to remind you that we need to think about nutrients or nutrition of the cow around parturition, not only in the context of the classical use of nutrients for a specific purpose, but also in the context of their functional capacity. And again, the best example of a functional nutrient that we know of is the role of methionine and choline in the context of one carbon metabolism. Lysine potentially can also be considered as a functional nutrient because of its role in the context of carnitine synthesis. But we also need to keep in mind that something like one carbon metabolism is not only going to rely on these methyl donors, but also on cofactors, trace minerals and certain water-soluble vitamins that are needed by those enzymes for optimal functioning of those pathways. There are some major targets that we need to continue monitoring in terms of of how the cow responds. Some are metabolic, some are non-metabolic, and among the non-metabolic ones are, for example, the immune response and the role of oxidative stress and inflammation in these overall adaptations of the cow. Remember, if we have healthy cows, they're potentially going to be better able to produce more optimally. And in the context of managing the nutrition of those animals for health and production, clearly the data seem to indicate that methionine and choline, potentially lysine, are also fairly important in the context of one carbon metabolism. Glutathione and taurine will give an additional advantage in the context of targeting pathways that we know are going to respond and produce, in this particular case, antioxidants that can help those cows remain in better health. Clearly, there's a lot more information that we need in order to understand how we can consistently fine-tune the nutrition of transition cows. There's a lot of interactions, a lot of other variables that often mean that we cannot have consistent results when we compare experiments or published experiments. So there's potentially some work that needs to be done in that area. And as I indicated before, we need to also start thinking more carefully about the potential benefits of feed additives, especially those that are related to microbial fermentation products, because there's some important data that are being produced, and I suspect there will be more experiments that will try to address the use of these microbial fermentation products in the transition period in the context of health and production. With this, I would like to thank everyone for your attention, and I hope this information has been useful.